Hello students, how are you all? I hope all of you are so amazing and you have seen all the recaps of the previous chapters. So today we are just going to start with the biotechnology and its application. So let's see the quick recap of this chapter in which we are going to see everything in a squeezed in a very sequential plus brief way, right? So when we talk about application of biotechnology, we know we use it in the medicine, we use in the diagnosis, we use them for genetically modified crops for agriculture, means we are just taking the gene from one plant and introducing it to the other plant, right? So that we can have the benefit of that, whatever the character that gene will be coding will be expressed in this particular plant, right? Processed food, bioremediation. So we know that nowadays these genetically modified bacteria are used to decrease the pollution because they are able to convert the petroleum or oil waste, oil refineries waste into the degradable one, right? So waste treatment, energy production. So these are, we have made the organism very efficient for all the activities from medicine to the energy production. So now we will see what are the critical research area in the biotechnology. So we know that they are providing the best catalyst to improve the organism, right? So suppose we are changing the genes of the bacteria, microorganism, making it so efficient for, for our use, right? So suppose we have put a gene which is forming a certain type of protein that we need to commercialize and we are going to get a lot of benefit, and this people will get benefit out of that product. So that's why we are using these transgenic animals to obtain the products, right? It can be enzyme or protein. So then we are creating all the optimum condition to produce the product that we have seen in the biotechnology and its principal chapter, right? And we are just purifying our product after it's get produced. We are just extracting it, right? So this is the main areas of research in the biotechnology. We are using these steps to get the product. Now we will see the agriculture, medicine, industries, and environment. So we are using these genetically modified or transgenic animals or organisms in different fields, in medicine, in agriculture production, right? So industries and environment. So green revolution, we know about the green revolution as our population were exploding or increasing. So we had to fulfill the demands of the food, right? So for that we brought this green revolution, right? So our many great thinkers brought this green revolution, in which we were using the chemical pesticide, means all the chemical fertilizer, chemicals mainly, to increase the productivity of the plant to fulfill the demands of the people, right? So that means it was having certain, this green revolution was using so much chemical that it was harming our environment in the long run, right? So that is the green revolution. It was done to to fulfill the needs of the growing population. But again, I have told you that it was actually degrading our environment and the good organism, the eco, it was, some way harming our ecological, balance, right? So to overcome this, we are having the other method, we introduce the other method for the organic farming. So we know in organic farmings, you must have done in the other chapters of plant breeding. So we are doing the animal organic farmings, in which we use the eco-friendly pesticide, biopesticide, or we are using the biofertilizers so that we can reduce the pollution, right? So we were using use of biopesticide, biofertilizers, etc., which are eco-friendly, right? So this is the eco-friendly, not causing harm to our population. So you know to overcome that huge chemical deterioration of the environment, we just introduce the organic farming. And then we have the genetically modified crops also. We are having the genetically modified organism now, right? So after the advancement of the biotechnology, we get to know that we are having another option also to increase the variety or to increase the production, and that is the genetically modified organism, or you can say GMO, right? So we are manipulating their gene, we are introducing the best gene which will be giving the best product to us in those plants, animals, microorganism, so that we can obtain the good product. So we have seen all the steps, how exactly gene is introduced in the different organism, what kind of vectors we use. So we already have done it in the principal process chapter. In this chapter, we are just seeing the application of that, right? So next is the advantage of genetically modified organisms. So we are just introducing the gene in the plant which is making them more tolerant towards the biotic and abiotic factor, right? So like most like more tolerant towards the cold, drought and salt and heat. So that means now the plant will not destroyed due to these reasons, right? And they will just keep on growing and give us good productivity, and that is the beneficial for the farmers also, no? Next is the reduced reliance on the chemical pesticide, because we are introducing the gene inside the plant that is making them pest resistant, so they don't need specially chemical pesticide to kill the pest, right? Next is help to reduce post harvest loss. Post harvest means suppose we have harvested plant or some fruits, and we know that if we are storing them somewhere, they are more they will just get ripened, and after that they will get spoiled. So we don't want the post harvest loss. So what exactly we will do, we will inactivate their gene, we will silence the gene which is responsible for their fast ripening. And what will happen because of that? Obviously we can store plant for the long duration. So that's why post harvest, and we will see the example of the post harvesting loss is flavor, savor, tomato. So yeah, we have tomato. So we have broad changes in the tomato, right? We have silenced the gene which is responsible for formation of the protein that is required for the silencing. Now, increased efficiency of mineral usage of the plant. So we have put such a gene which is making them more efficient to absorb the nourishment or whatever nourishment is there in the soil. No matter if it is having the low nourishment, but still they become so efficient to absorb it now, right? And next is enhanced nutritional value of the food. Example, golden rice. So this is the best example of biofortification. Biofortification means we are enriching the nourishment level also in the plant, right? So we will see how exactly. So the best example is golden rice that we are going to see. This is the golden rice. Rhiza sativa is the scientific name of rice, no? So what exactly we have done, how exactly this biofortificated, or you can say this vitamin A rich rice was formed, right? So transgenic variety created by using gene beta carotene. So we are using the beta carotene gene from the daffodil plant. So there is a gene in the daffodil plant, daffodil plant. So we are taking the beta carotene gene from it. We are just cutting, from the principal part. We have seen how to cut the gene from the plant cell. So we have taken the DNA, cut it with the restriction enzyme, then we separated it on gel electrophoresis. So we have done all the steps in principle. I'm just telling you the application of it, right? So we have isolated the gene for the beta carotene from the daffodil plant, right? And now we will paste it with the TI plasmid of agrobacterium, and then we will put it in the agrobacterium, let it infect the plant, right? So what is the purpose of this vector TI plasmid? We know it will introduce our gene of interest, that is the beta carotene, in the plant cell. And now this plant cell will grow to form the huge plant, and golden rice will give the golden rice, right? So now we are introducing with the help of the agrobacterium into the rice. And now this rice plant will grow up to form the golden rice because it's rich in the provitamin A. And because of the beta carotene gene, it is forming certain pigment also, yellow pigment. So that's why it look like golden rice. And that beta carotene is having the provitamin A. And when a person consume this golden rice, which is having the beta carotene, which is having the provitamin A, will be converted by liver into vitamin A. And you know the purpose of vitamin A? It prevent the It cause is basically prevent the disease night blindness. So we know that night blindness deficiency caused by the vitamin deficiency of vitamin A. So it will prevent night blindness, right? So that means it is increasing the night blindness. It is increasing the nutrient value, that is the nutrition value in the plant, right? So vitamin A enriched plant is the golden rice. So I hope you all get to know that how exactly we are doing the biofortification of the golden rice using the gene from the other plant known as daffodil plant. No, next is the golden rice. We have seen, we will see the bt cotton, corn, brinjal, even we have from tomatoes, we have from potatoes, bt, right? Bt tomato, bt potato, right? So we have bt rice, corns, etc. Pest resistant tobacco plant, how to make the pest resistant, we will see. Virus resistant plant, flavor saver tomato. So now we will see the flavor saver tomato, which will prevent the post harvest loss. So we can preserve them for the longer time and they will not spoil, right? So let's see about the flavor saver tomato. So this is the tomato, and the cell of the tomato have a gene, right? It is having DNA. DNA have a functional part of the gene which is coding for the polygalacturonase enzyme. And this enzyme is responsible for softening or ripening of the fruit, right? It will degrade the pectin, pectin wall so that they can become so soft. So now what we have done using the RNA interference technique, what we do, this gene, obviously we know that what happened generally, gene transcribed into mRNA, right? And using RNA interference, we are modifying this tomato plant in such a way that this mRNA having another and complementary RNA also, right? And it is converting into double-stranded RNA. So once mRNA converted into double-stranded RNA, it will become inactive, will not undergo any translation, means it will not form PG enzyme, means polygalacturonase enzyme will not be created in the tomato. And now when it is blocked, we can store, the ripening process will be slowed down and we can store it for the longer duration. And this is how we can actually prevent the post-harvesting loss, no? So isn't it amazing? Isn't it amazing, kids? So this is how normal tomato look like. It is having PG gene, which undergo transcription to form mRNA. mRNA undergo translation to form the polygalacturonase enzyme, and this enzyme will degrade the pectin and it will convert into the ripened fruit. So when we talk about flavor saver tomato, what we are doing, we are modifying the tomato in such a way that it is having the complementary RNA strand, complementary mRNA strand for this mRNA, right? So this pgDNA will form the sense mRNA, but we are having the stored antisense RNA. So as soon as the sense RNA is formed for this enzyme, what will happen? We have modified this tomato plant to create the antisense strand, which is complementary to this mRNA. And then obviously it will block the translation. No enzyme, no PG enzyme means now the ripening will be delayed. So delay in ripening, prevent post-harvesting loss, and flavor saver tomato can be stored for longer duration. So you're having very, very beautiful notes. You can just preserve it for your exams also. You can keep it with you. Now we are talking about the Bacillus thuringiensis. So when we talk about Bacillus thuringiensis, what exactly happened in this condition? So we know that Bt cotton. What is the Bt cotton? It's a biopesticide plant. It is having a gene, means we have introduced a gene in the cotton that it is actually producing the pesticide naturally from the plant, right? And it is killing the insect caterpillar so that they will not spoil the cotton. So some strain of Bacillus thuringiensis bacteria, it's a bacteria, is producing certain toxic protein, and that protein kills the insect, right? So this bacteria has that gene which will code for the toxic protein, and that toxic protein will kill the insect. So like what kind of insect? Lepidopterans having all these insect, Coleopterans having the beetles, Dipterans have the flies and mosquitoes. So we are having the gene in the bacteria to kill all these kind of insects that I have written over here. So now what happened? This is the Bt bacteria, Bacillus thuringiensis bacteria. Bt from the Bacillus thuringiensis, right? So it's a bacterium, and now it is having the DNA having a special gene known as cry gene. Now you will say, ma'am, cry means crying. No, cry means crystal. So gene which will code for the crystal protein, right? So this cry gene is endotoxin gene. It will form the endotoxin. Toxin means poison, right? So it will convert into mRNA, and this mRNA will convert into crystal protoxin, means inactive protein, inactive toxin form. So in bacteria, this protein is formed in the crystal form. Cry means crystal. And this crystal is inactivated. So means it is not harming the bacteria, right? But suppose this is the leaf of plant. And the leaf of plant has these Bt bacteria on it, sitting on it, chilling, pilling. And this insect are eating the leaf, and leaf has these bacteria, and they started eating such bacteria also, and these bacteria has their defense system. They will be like, oh, I need to save myself from this insect body, so I will kill this insect. How this bacteria will kill the insect? So they will enter the midgut of the insect, and in the midgut of the insect has the alkaline pH, and that alkaline pH will dissolve the crystal into the soluble form, right? It will dissolve the crystal and convert it into the active form. So now they convert activation in alkaline medium, means this alkaline medium will dissolve cry protein, protoxin means which is inactive form of protein. It will dissolve cry protein and it become active, it become toxin now. And once it become active, it will bind with these. So suppose this is the gut of the insect having the receptors. So it will bind with the, attach with the epithelial wall, epithelium of the midgut, and cause the lysis and pores on the epithelium cells of the gut. And gut has the pores now, has the, this lysis, the cells are disrupted and cause the pore in the gut, and all the food is leaking in the cavity, it's not reaching the stomach. So due to starvation, this insect will die. So this is how exactly this mechanism works, right? So same way, we have used this cry protein as a pesticides. So we have got to know that, yeah, this bacteria has a beautiful gene, right? So suppose this is the bacteria, right? And then bacteria has this gene known as cry gene. We have cut it with the restriction enzyme and we just release it out. So suppose this is the cry gene for the crystal protein, right? Now this cry gene will be introduced in the TI plasmid, Agrobacterium TI plasmid. So we are introducing this in the TI plasmid. So let's paste them together. So we'll cut it with the restriction enzyme, paste cry gene here, it become recombinant DNA, and using agrobacterium now, right? So using agrobacterium means agrobacterium bacteria, right? Agrobacterium mediated transfer. Using agrobacterium, we will be transferring it in the plant cell. So we have introduced this gene in the plant cell. So suppose this is the cotton plant cell, right? So we have introduced this gene with the, suppose this is the gene of the plant, and we have introduced our gene also, right? Our Bt gene also. So now this plant is known as Bt cotton. So it has converted into Bt cotton. When it will grow up, what will happen? The same mechanism will work here. So suppose insect will eat it, and in the insect gut it become activated, no, and once this toxin become gets activated, it will kill the insect. The same mechanism will follow because now it is having the Bt gene to form the cry protein. So what kind of genes are there? Types of the genes. So you have to remember protein encoded by the genes cry1AC, and this is the other gene of the cry which will control the cotton bollworm. So cry1AC, cry1AB will control the cotton bollworm, and the gene cry1AB will control the corn borer, right? So 1AC, 2AB cotton bollworm, 1AB corn borer. So you have to remember this for your NEET exam, right? So these are important. So now let me take you ahead all about the BT cotton. So now we are going to discuss the pest-resistant tobacco plant. So nematode Melidogyne incognita lives as a parasite in the root of tobacco plant and infect the root and decrease the fertility and decrease the production yield of the tobacco plant, right? So this is the roundworm. We have to make this plant resistant to this parasite, right? And plant is made to resistant to infection by using RNA interference technique. So I have discussed this earlier also in flavor saver tomato, but here I will elaborate the RNA interference technique, which confuses many kids. So if you read NCRT lines, these NCRT lines might confuse you, but you listen to me carefully, everything will be clear, right? So now you just see, let me show you this page. RNA interference, or we also called it post, means after transcription. You know what is transcription? When mRNA is formed. So after formation of mRNA translation, we will not let the translation of the toxic gene of nematode happen. So Melidogyne incognita is a nematode which will be causing infection in the root of the tobacco plant, right? So we have to prevent the formation of the toxin which is responsible for that infection. So obviously that toxin is formed by a gene. So we are targeting that gene which is causing the infection in the plant, in the nematode, right? So suppose this is the gene of the nematode which is forming mRNA. mRNA forming the protein or protein in nematode. So suppose this is the nematode specific gene, right? Nematode specific gene. And this protein in the nematode is infecting our plant. And this is responsible for the root knot disease in the plant, in the tobacco plant, to reduce the productivity. So what we have to do, we have to specifically target this gene, nematode specific gene. This is the specific gene responsible to form the toxin which is creating the disease. So we have to target this gene. We have to convert this mRNA into double-stranded mRNA so that we can prevent the translation of it. Post-transcriptional silence we have to do, right? We have to interfere with this RNA because this is the dangerous one, right? Now, so Arabidopsis model plant was the first plant where we have seen this experiment. Where we have done this experiment, RNA interference. Our scientists have done that experiment. So RNA interference is present in all the eukaryotes as a regulatory mechanism. Means it is silencing certain genes in, those the genes whose expression we don't want, right? And it is also act as a defense mechanism in eukaryote. So defense mechanism, suppose there is a viral infection in the body. So we are having this RNA interference, which will form the complementary bond with the mRNA of virus and it will prevent the infection. So you can say that it will give us defense against the virus also, or the eukaryotes have this defense system in their body already present. So RNA interference is not new, it is already inbuilt in our body. So inspired by that natural mechanism, we thought that why not to use it in the biotechnology in order to prevent certain infection. So that is what we are doing. So our purpose here is to convert RNA into double-stranded RNA so that it become inactive, and no translation means no toxin will be produced. So this gene, suppose responsible to produce the toxin, now that toxin will not produce and plant will not be infected. That is our main aim, right? So this is the nematode specific gene, which is the source of. So what is the source of double? So we are just talking about the biotechnology, how we are using RNA interference in biotechnology. How we are making the tobacco plant resistant to this gene or this toxin. Let's see. So suppose this is the nematode specific gene. So we required the gene which will, we required the same sequence of the gene which will actually create the double-stranded RNA in the plant cell. Right, when this gene, trying to infect the plant, when this product of this gene trying to infect the plant, we need complementary mRNA of the sense RNA, sense mRNA, because it will form the sense mRNA and which will undergo translation. But we have to silence that mRNA. So we are just seeing the sequence of the gene and modifying virus RNA, a retrovirus RNA, modifying their genetic material for our purpose, which is specific to the nematode. Means it is same as the nematode, right? Or specific targeting to the nematode gene. And we can use transposon also as a source of double-stranded RNA, right? Because our purpose is to form the double-stranded RNA to protect our plant. These are the two sources we can use as a source of double-stranded RNA, right? So using the genetic material, we are taking the nematode specific gene, putting it in the TI plasmid and making the recombinant DNA. Right? So using the agrobacterium, now we are putting this gene, specific nematode specific gene, which will form the double-stranded RNA. We will introduce, using agrobacterium vector, we are introducing it in the tobacco plant, NEET line, right? So NCRT line, and important for NEET. So agrobacterium is used to infect, or we are just, this is the vector, right? So this vector, having the nematode specific gene, and it will introduce it in the tobacco plant, right? So this gene will be introduced in tobacco plant. And now tobacco plant will show the RNA interference. How? Let me show you. Now this plant will grow up and show that effect. So this is the plant cell, which is having the nematode specific gene also, right? So suppose this is the nematode specific gene. And this gene is forming the double-stranded RNA because we have modified viral RNA for this specific purpose, right? So obviously this retroviral RNA will be pasted with the plant gene and form this double-stranded RNA now. So this double-stranded RNA having one sense strand, other one having the antisense strand. So there is enzyme called dicer, which is the RNase 3. The enzyme is RNase 3 called dicer. Dicer means it will diced the RNA, right? This double-stranded RNA into small interfering RNA, siRNA. So siRNA is small interfering RNA. Now we are having the other enzyme complex known as RISC, RNA-induced silencing complex, right? And along with the helicase, what will happen? Unwinding of sense and antisense. So sense strand of RNA will be separated from antisense. They got unwind. So what will happen after that? Sense strand will be digested by RISC. So that means sense strands are digested. These are digested, and antisense strand, which will be complementary to the mRNA of nematode, right, will be stored in the body. So now it is stored in the plant cell. So suppose now we are having antisense strand stored in the plant cell with the RISC protein, right? So these are the antisense strand. All these are the antisense strand in the plant, right? In the plant. So suppose this is the nematode which is infecting the plant. So nematode has a gene which will form the mRNA, right? So it is the sense mRNA, but we have already stored complementary mRNA in the plant body. So this sense strand will form the this antisense strand will form the complementary bond with the sense strand, which is produced by the gene and responsible for the formation of protein, toxic protein. So what will happen? Now double-stranded RNA is formed. Double-stranded RNA is formed. We have done RNA interference. No translation will happen and no toxin will be formed. And we have protected our tobacco plant. So I hope this concept is pretty much clear to everyone, right? So this is the summary. I have told you that double-stranded RNA, which will be formed in the plant cell, cut by the dicer into siRNA, small interfering RNA. Then RISC protein and helicase will separate them, and RISC will digest the sense strand. And then when the nematode infection happen, so nematode will form the antisense strand, and we are having complementary antisense strand in the plant. It will form the double-stranded RNA and no translation will happen. So this is the summary. So that's all. This is how exactly RNA interference will be done, and we are protecting our plant from the root knot disease. So now it will not be infected by the nematode, no? So next is the human insulin. So we know that naturally when we talk about human insulin, so let's talk about it, right? So what happened in the human? This is the structure of the natural insulin, right? So insulin is produced in this form, known as in the form of proinsulin. So our body produced pro-insulin, means inactive insulin, and then it is converted into active insulin. So what is the difference between active insulin, this is the proper insulin, and what is the difference between pro-insulin, right? So pro-insulin has A chain, it is having B peptide chain, means it is made up of protein, right? Amino acid. So insulin is a proteinaceous hormone which is made up of amino acid, right? So now if you see this, we are having A chain, B chain, and C peptide chain. So this C peptide chain will be removed from the A and B, and A and B are connected with the disulfide linkage. This is known as insulin, and mature insulin don't have the C peptide. So this is the mature insulin, proper insulin, which doesn't have the C peptide. It doesn't have the C peptide, right? So this is the structure. Now we are just going to start with the gene, this insulin gene, how it is produced. So when we talk about the insulin, I have shown you the natural insulin. So a lot of issues were there to produce the insulin because, A chain, it was very, very difficult to produce A chain, B chain, and then, to link them with the disulfide bond to form the insulin. So finally, there was a company known as Eli Lilly, American company, in 1983, right? So they were able to produce the insulin with the help of gene. So we will see how exactly they have done it. So insulin is used for treating the diabetes. We know it will control the blood sugar level. It will lower down the blood sugar level. And insulin earlier used to be extracted from the pancreas of cattle and pig in the slaughterhouse. But these, insulin of these animals were creating the allergy in some humans. So many companies tried to create the insulin with A and B chain with disulfide linkage, but they were not able to produce them. So finally, this company, we will see how exactly they are doing that, how they are producing the A chain B, how they are producing all the artificial insulin, right? So what they have done, this Eli Lilly company, right? So this company, American company, they have artificially synthesized gene for the A peptide and artificially synthesized gene for the B peptide. Mark my word, right? So they have synthesized, they have synthesized A and B chain, B gene artificially, because they have seen the sequence and artificially synthesized the DNA. So now what they have done, suppose this is the A gene and this is the B gene for the B peptide chain, right? So they have for the A peptide chain, and this is the B gene for B peptide, right? So they have used the E. coli here. So suppose they have used this E. coli in this medium. So this E. coli in this medium, and they have just used the other set of E. coli in other medium, right? In which they will grow A gene, and in other they will just grow the B peptide. So these gene will form the peptide, A peptide, B peptide. So they are having this plasmid, and we have pasted our A gene with it, and we have pasted the B gene with it, right? So this is the B gene and this is the A gene. Now we will let them grow, right? This is the E. coli. NEET question, that what is the vector used here to produce the humulin? It will be, humulin means artificial insulin, right? So it will be E. coli. So E. coli will keep on growing in the bioreactors, and it will keep on producing the A peptide. So it will produce the A peptide chain, which we will separate by downstreaming process, and it will produce B peptide chain. So B peptide chain, mRNA and then protein will be formed, peptide chain will be formed. So this is the A peptide and this is the B peptide. Now they have pasted A peptide, they have separated A peptide and they have separated B peptide, and using disulfide bridge, they have connected them. So in this way, they formed the proper humulin. So this is how they have produced the humulin or genetically engineered insulin. So you have to remember all these steps, that what they have used and how they exactly have created it, right? That's all. Now we will see the gene therapy. Gene therapy means we are correcting the defective gene of a person or mutated gene of a person. So permanent cure of the permanent cure for the gene therapy is done at the early embryonic stage only. After that, we don't have the permanent cure. We can actually do the regular methods, some regular therapies, right? So when we talk about normal genes are delivered to the embryo of an individual with a faulty gene, means we are replacing the faulty or mutated gene with the normal gene. So suppose early embryonic stage having the mutated gene in the early embryonic stage. So in the early embryonic stage only, because in the early embryonic stage we have few stem cell, and they are not differentiated so far. So it's easier to put the gene there, and it will be expressed in the whole embryo when it will grow up. So we can just permanently solve this mutated gene problem, replace it with the normal gene only at the very, very early embryonic stage, not later. Later, we don't have any permanent solution, right? So it will be replaced with normal gene. Replaced with normal gene. So we can permanently cure it only at the early embryonic stage. Here we can permanently cure it. It will be permanent cure. But after the later stage of embryo, when the differentiation of the tissues is started, when the differentiation of the cell is started, then we cannot do any permanent cure, right? Then we have some temporary cure that we will see. So first time gene therapy was performed on a girl who was having the enzyme deficiency known as adenosine deaminase enzyme deficiency. And this enzyme, ADA gene, ADA protein, ADA enzyme, right? Adenosine deaminase enzyme, adenosine deaminase enzyme is basically responsible for maturation of lymphocyte, maturation of T lymphocyte. And we know that T lymphocyte is helping in overall immunity. So when there is the lack of, the gene which is responsible to form the ADA enzyme is mutated, what will happen? In that case, no ADA gene will be produced, no ADA enzyme will be produced, and no maturation of T lymphocyte. And when there is the no maturation of T lymphocyte, what will happen? No maturation of T lymphocyte means they will die, right? They will just, immunity will lower down. So it will cause the immunity deficiency or severe combined immunodeficiency disorder, because of the decrease in T lymphocyte, this B cell also will not give any strong response. So overall combined immunity means B cell, T cell, both will lower down, right? Both will be affected here. So ADA deficiency caused due to deletion of the gene or mutation of the genes coding for ADA. Functional ADA complementary DNA can be used into, so we will see this, part. So I have told you that it is permanently cured only during the early embryonic stage. And the other process, which is the temporary stage, temporary, process. So temporary solution, temporary treatment is the enzyme replacement therapy. Means we can we can inject the enzyme in the person, ADA enzyme in a person, enzyme therapy, enzyme replacement therapy. So we are just injecting the enzyme, ADA enzyme in the organism, in the human who are having the ADA deficiency. Or we can do the bone marrow transplantation, bone marrow transplantation. Or we can do the gene therapy also after birth. But these are temporary stage. After birth or after later embryonic stage, it cannot be permanently cured, right? So we will see that how exactly gene therapy is done. So suppose this is the patient having the ADA deficiency, right? Having ADA deficiency. And for the first time, it was seen in the 4-year-old baby deficiency. So what we will do, we will just take out the T lymphocyte of this patient, because T lymphocyte require this gene. So we will just take out the T lymphocyte, right? So we have taken out the T lymphocyte which have the mutated gene, and using the retrovirus vector, right? Retroviral vector. In animals, to transform the animal cell, we need the retroviral vector. And you know what? This vector will be carrying the good gene, right? Which will be carrying normal ADA gene. Normal ADA gene. So it will be replaced with the faulty gene. So this is our T lymphocyte. We have growing the T lymphocyte. We are taking the patient T lymphocyte out and then we are growing it in culture, right? We grow them outside the body so that we can just take the normal gene. We can just introduce the normal gene in their T lymphocyte. So these are the recombinant T lymphocyte or T lymphocyte with the ADA gene. So T lymphocyte with the ADA gene. So we again take it back to the patient body, right? But this process is required periodic process, or it's a periodic process, because T lymphocyte is not immortal. They are not like living for throughout the life. They have certain lifespan. So after that they will be dying, right? And we have to require periodic infusion of this process. So it requires the periodic infusion, right? So we require periodic or repetition of this process because they are not immortal. They are not immortal. They will die after some time. And then we need to replace them, or we need to do the periodic transfusion of this process, right? So this is the replacement therapy. So that's all. Now we will see the molecular diagnosis. So molecular diagnosis. So traditional diagnosis, when we used to diagnose the disease, seeing the stool test or urine test or blood test. So these are the traditional method. They used not to tell us about the disease at the early embryonic or early stages, right? So it will give us the idea only at the later stage or advanced stage of a disease, when it will become so difficult for the person, for us to save the patient, right? So these molecular diagnosis, advanced molecular diagnosis, include PCR, ELISA. So these techniques are so efficient for us to detect the disease at early embryonic stage, right? So we can detect any disease at early embryonic stage using the two process mainly. So we can detect disease at early embryonic stage. Early embryonic stage. Oh, sorry, at early stage, right? Early stage of disease. So PCR machine, we know that suppose a person having AIDS, they must be having the mutated gene in them. So what we are doing, we are just amplifying. Using PCR, we are amplifying the gene so that we can actually study it very nicely. By amplifying it, so that we can study very nicely that if mutation is there or not. So means PCR technique is used to amplify the gene which is which is the suspected gene. We feel that this gene can be mutated, right? So very low concentration of virus and bacteria can be detected by amplifying their DNA, used to detect the HIV virus in the AIDS patient, and mutation of the gene in the cancer patient also. So we are actually amplifying the gene to make this study very nice, or very significant for us to know that this person having the mutated gene. Enzyme linked immunosorbent as a ELISA. So what is the ELISA? What is the technique behind, what is the principle behind this? Antibody antigen interaction. So suppose a person having a disease, we know that this person must be having this type of antigen, or this person must be forming this type of antibody. So extracting the, because when we talk about cancer cell, so cancer cell have a specific antigen. So we will just have the preformed antibodies in the lab. We will take the suspected cell and we will just put the antibody there, which is specific for the cancer antigen. So when antibody stick with it, we will get to know that this person having the cancer. Same, a person having certain disease, they will be having certain antigen in their body. So if we are suspecting that this person having that antigen, that antigen of that disease, So we will be using the antibody to see if it will be sticking with it or not. If it stick, we get to know that yes, if antigen antibody are sticking with each other, means this person having this disease. So these are the diagnosis disease. Then we are having confirmatory diseases, sorry, confirmatory methods. Confirmatory methods means how to confirm that this person having this AIDS and all. So we are having the blotting, northern blotting, southern blotting, western blotting, right? So western blotting used to detect confirm the AIDS also. So these are the diagnostic test, and other blottings technique are the confirmatory test. So this was the NEET 2021 question also. Don't get confused, what are the molecular diagnosis method, right? So you know PCR amplify the gene, which we have done in the last chapter. So now we will see the transgenic animals. So we know that uh, why we are using the foreign gene and introducing it in the animals, and 99% transgenic animals are mice. We know very well 95% transgenic animals are mice, because mice are easily available. They have shorter breeding period, and they will be able to produce a lot of babies. A lot of results will be studied. So study of gene regulation, how gene work, how its normal growth and development happen. So suppose we want to study the function of gene, we will use the transgenic animal for that, right? So production of various biological product like protein, certain protein are produced like antitrypsin. Antitrypsin protein, so we are just introducing this gene in the sheep. We have introduced this gene in the sheep, and this sheep was producing antitrypsin, and which is used to treat the emphysema, right? Rosie. Rosie is the first transgenic cow which is having the gene for human lactogen. So we have taken the gene from human, introduced it in the zygote, and this zygote grown up to form the Rosie, and Rosie now forming that gene, that protein that from the gene known as alpha lactoalbumin, right? So which is the milk of this Rosie cow having the more protein in it, right? More this protein, 20 gram per in that ways, right? So it is having the more protein content. So safety of vaccine can be tested, example polio vaccine. So before selling it in the market, we test any efficiency of any vaccine, or if it is having any side effects, we just test it in the animals. And these animals are actually becomes make so sensitive for these vaccines, so that they will be showing all kind of reaction. Even in a sensitive person, we can just see using these transgenic animals. Toxicity of drugs also checked in the transgenic animals, right? So these are the different transgenic animals. Now we will see the organization GC, Genetic Engineering Approval Committee. So this organization will approve that this biotechnologically modified organism is good to use or not by the public, right? So that will be done by the GEAC organization. We know the story of basmati rice. I'm sure you also know the story of basmati rice by piracy. So it is grown in our country from decades, or from since the ages. But the American company was selling the basmati rice with their name. So they were doing the buy piracy without our consent, without our permission. They were just selling it with their own name, right? So when we got to know, so there are many great activist people, so many advocates are there who actually won the case. And then we got to know, I mean, we have taken their patent back, right? So we were like, no, this is not your resources, this is ours. So 2 lakh varieties of the rice are there in India alone. Basmati rice is having the distinct aroma, and 27 documented rice are grown in India. Documented means we can see it in the documents. Our traditional folklores, our traditional books have mentioned all these things. 1997, American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad with their name itself. And new variety of basmati actually derived from Indian farmers. And Indian basmati was crossed with the Swami dwarf, claimed as and mentioned a nobility, means their new research. But it was our research, right? It was our farmers, resources. So several attempt has been made to patent uses, products and process Indian traditional herb medicine. They have just, they were using the turmeric, neem, which are used in Indian traditional, it is mentioned in the folklores or in, again, our traditional books. So we are using it since ages. And they were selling all the products made up of these, or they were selling the neem and turmeric in the market with the name of their company. So means they were actually exploiting our resources, right? So that is the biopiracy. Biopiracy means they are using our bioresources without our consent, right? Without any compensatory payment, they are just using it, saying that this is our discovery, right? So that is the biopiracy. Biopatent means you are just patenting it, you are taking the copyright, or you will be giving the when you are going this when you are patenting something, means you are having the rights to use it, and if anybody want to use it, they will take permission of yours, right? So that is by patent. So that's all. These are certain questions for you, so you know the answers now. You know the answers you will be solving because you are getting the PDF. You are getting the PDFs. Thank you so much, kids. I hope you find this chapter so amazing, and you are seeing all the lectures that we have covered so far. Have a great day.